kind of catch us up with the state of the art than Dr. Margaret Liu. She's a researcher who studied the immune response in vaccines for many decades, is often known as the mother of DNA vaccines, and has studied DNA and RNA vaccines for many years, and is going to help give us a bit of an overview of where we are in the vaccine space now and going forward. So Dr. Liu, take it away, over to you.
Great. Thanks very much for the invitation to speak and to talk about vaccines. And one of my hopes is that even though I think that a vaccine may be quite a bit of time in the future, at least a year, maybe 18 months or more, that this certainly will help people appreciate the whole notion of protection, that is, before people get the disease at all, and also try to help us become more prepared for future pandemics. So what are the challenges for making a COVID vaccine? Well, just the first one that I mentioned, which is the time required to make a vaccine compared to the speed of the pandemic spreading and where we are now being already behind the pandemic. One of our big challenges is that we don't really know exactly what type of protective immunity we need. I think that Divya gave a great presentation in terms of talking about the aspect of binding to the spike protein of the coronavirus, binding to the receptor, the ACE receptor that we just heard about from Brennan. But the challenge is that we don't actually 100% know that this will be protective to be able to block that binding. So which parts of the virus do we target to make a vaccine is not known, although at least we have a good place to start. And one of the challenges is that usually for making a vaccine, we try to first go into animals and try to see whether or not a vaccine that we make can be protective. So I'll talk more about that as we go on. But one issue I want to raise from the beginning, because if you've been reading any news articles about vaccines, you'll know that there is the possibility when you make a new vaccine that you have to make just the right type of protective immunity. Because if you make certain kinds of antibodies, it is possible for certain diseases that these could actually make more infection rather than less, or could worsen the disease severity. So it's very important, even though vaccines have been the single most important medical intervention and have actually eliminated the disease smallpox from the whole planet. It's also eliminated another disease called rinderpest from the planet. It's an animal disease, but there have been 2 diseases eliminated by vaccines. But you have to know exactly what's the optimal type of vaccine to develop. So we saw some lovely examples of the spike protein, and how here the idea is you can see these antibodies are these Y-shaped proteins that the body uses to, for example, bind to a virus, and then perhaps by binding to the spike protein, that will prevent the spike protein from binding to the ACE2 receptors. So this has been the first target that people are using. Yeah.
Your slides, make sure you hit the share button, because we're not seeing your slides.
Oh, you're not seeing it? Oh, sorry. I thought I did hit the show button. Let me go back then to that, and I apologize for that. Sorry about that. Let me go back here. I was trying to get all ready just as Brendan finished, and instead okay, let me just gonna share stream. Here we go. How is this now? Are you got me? Is that showing up now?
Yep, there we go. Perfect.
Okay, great. Sorry about that. So we're gonna, what I'm going to do though is, you actually had seen already the great slide before that had shown the antibodies binding to the receptor so that it blocks binding of the spike protein. One of the challenges, however, is that in the earlier SARS virus number 1 in 2002-2003, that patients who had actually had antibodies to that infection, it turns out they made antibodies to a lot of other proteins. Now, those antibodies maybe didn't help prevent the virus from binding, but it does show that there may be other proteins that are really important when you make a vaccine. So because we did already have one SARS epidemic before that was from a related virus, that was SARS-CoV-1, as well as the MERS, the Middle East Respiratory Syndrome, what can we learn from those previous efforts in order to make the vaccine now? Because there were many vaccine efforts, and they actually even some of them went all the way to human clinical trials. The problem was that the inactivated covirus, for example, that was tested then made great neutralizing antibodies that if you tested the antibodies that you got from an animal and you tested against the virus in a test tube, it was great at neutralizing the virus. But the animal studies actually had less compelling data for protection. And then, fortunately, the epidemic ended so soon that there were no clinical trials that actually went on enough to be able to show protective efficacy. So the challenge is that from those earlier studies, we don't yet know from SARS-CoV-2 whether or not using the spike protein will be, in fact, as efficacious as we want. So we don't really know, are antibodies going to be enough, and will a spike protein be the best target, even though it makes a lot of sense to start there. So I just want to give you a little brief bit about different types of virus vaccines, because in fact, you'll see from what I show later on that there are now 34 different vaccines being developed, and they actually use almost all of the different kinds of vaccines that you see on this chart here. And I just want to make the point that in fact, the first 5 that you see have all been used successfully. These technologies have all made various vaccines that have been used and licensed either for humans or for animals. So only the bottom one, the messenger RNA vaccine, which in fact is the one that's been in the news most, is the one that has not yet been licensed for any human application or animal application. So, but all of the other technologies actually have been very useful for making vaccines against a variety of diseases, and actually have helped to, eliminate and eradicate diseases in at least certain countries. Not all of them have been eradicated, but certainly have been very effective. This is a cartoon that shows you, if you want to think about what are these vaccine technologies. If we, in the middle, this drawing is actually of an HIV virus. But it's very similar in the sense that it does have the surface proteins that are kind of like the spike proteins. And so the technologies I'm going to talk about are the 5 that don't have the X's through them, and that's a whole inactivated vaccine. So basically here, you just grow up a lot of the virus and then you kill it somehow, usually with a chemical like formalin. And then when you give it to the individuals, it looks kind of like the real virus, but it's not alive, so it won't cause disease, but you can now make at least an antibody response against the outside. Now, if you didn't want to have to give the whole virus, and maybe part of that's because you don't want to have to grow it, then what you could do is you could recombinantly make just a single protein or various proteins that you want from the virus. And so that's what a recombinant subunit virus is at the bottom of the screen. And that's basically in a vessel, a vat, in cells, you can manufacture just the protein you want through recombinant technology. I'm going to go ahead and talk about these vaccines, and I'll come back and explain the others. So inactivated viruses have been used for various vaccines, including many of the influenza vaccines that we get every year. And so it's a tried and true technology. The challenge with it, and the reason it isn't one of the first ones in, is that there's a long development time, because you have to figure out how do you grow up all of this virus before you inactivate it. And then how do you make sure that you've completely inactivated that virus? So you could imagine that if you were working in a factory that was producing a lot of live virus before you inactivated it and killed it, that would be a challenge to protect the workers and to make the right process. So it's a very tried and true technology, and it does include a lot of other proteins. So that may be a good thing if those other proteins are good for making an immune response, but it's not clear that we don't know enough about the SARS coronavirus 2 yet to know which ones to include besides the spike, if the spike is useful. Now, since the spike is the major protein that's involved with binding to the human cell receptors, it would be a good approach to potentially recombinantly make just this protein. So you manufacture it in vitro in a vat that you put, then you have to purify it. And then because usually these proteins alone don't cause enough of an immune response. You have to find an adjuvant or some other chemical that will help kind of inflame the immune system as you inject this protein. So once you decide which protein to use, which in this case, the spike is a good one to start with, then you can make this. Now, again, it's a relatively long research and development process because you have to figure out what cell you can make the recombinant protein, and you have to figure out how to purify it and so on. Now I'm going to take a step back and just tell you a little bit about immune responses, because so far I've talked just about on the left about antibodies, these Y-shaped proteins that can directly bind to the virus and essentially can kill the virus by various mechanisms or can prevent it from binding to and infecting a cell. There's a whole nother arm of the immune system called the T cell arm. And in this case, there are one type of T cells that are called killer T cells. Now, these cells actually can't kill virus directly, but what they can do is they can kill cells that have been infected by the virus. And when they do that, they're essentially shutting down virus factories. So they won't prevent you from getting infected, but what they can do is they can help prevent you from getting a lot of more virus as the virus takes over your cells and starts to produce progeny virus to infect other cells. So it may be that we need to have both kinds of immunity. And that's where some of these other technologies may be able to play a role. Now I'm going to go back to some very basic biology that I'm sure all of you know, but as you know, we all have DNA in our cells, and the DNA is like our basic blueprint. But in order for the DNA to tell the body what kind of protein to make, the DNA itself has to be copied into another kind of molecule that's RNA. And in this case, messenger RNA. It's called messenger RNA because it's the message that's transcribed from the DNA that then enables the proteins to be made. So before, people always thought that you needed to give, for example, the protein, that DNA and RNA in and of themselves wouldn't be able to be used as vaccines. And that's for a number of reasons, but mostly that they're both pretty easily destroyed. And so it was thought that you couldn't just stick it into a syringe and inject it and the cells wouldn't take it up. But in fact, it turns out that scientists led by Phil Feldner years ago at a company called Vical found that just as a virus can bind to a cell and infect it, just like the SARS coronavirus can, that it turns out that even just a circle of DNA called a plasmid can actually, it turns out, be internalized into certain cells. And so people now, this is what my group then working with the Vical group had demonstrated, was that you could in fact take a circle of DNA, have it code for a protein from influenza, and it turns out if you just inject that into animals, or now we know in humans, you can then get an immune response against the protein from the influenza virus and many other pathogens. This has now been in many different scenarios. So it turns out that in fact, you can just use this as a vaccine itself without having to have the whole virus and certainly not all the other viral proteins. More recently, there's been increased effort on mRNA, messenger RNA, and that's because originally messenger RNA is much more unstable than DNA is, and it's also more toxic. It causes a lot more inflammation. So a number of groups have actually figured out how to make synthetic RNA that can be made very rapidly, and therefore this is why this is the first vaccine that was made that started human trials for COVID, and that's because it could be very rapidly made simply by knowing the sequence of the virus itself. And it's right now in a phase 1 study, which means that it's simply a safety study. They will also measure immune responses against the spike protein, but it's predominantly to make sure that this is a safe technology to use. Now, one of the challenges for this technology is that because the messenger RNA itself is inherently unstable and is actually causes a lot of inflammation, that the companies, several companies doing this, have had to develop proprietary technologies to stabilize the RNA and try to make it a little less inflammatory. So as I said, there's no RNA that has made it into any licensed products, but it is a promising technology, and partly because of its speed, but it's one that does have a lot of proprietary issues around it. So it may be a challenge in terms of manufacturing, even if it does prove to be effective, but at least it has gotten me into the clinic quickly. DNA vaccines likewise can be very, very rapidly made because it's the same thing. All you need to know is the sequence of the virus. And this technology has been shown to generate both the antibodies and the T cells. So if you do need both, they can both be generated. It's known to be a very safe technology because it's actually been in thousands of patients, of human patients, and for many, many diseases, both prophylactic vaccines and for cancer and autoimmune diseases and asthma. There are actually several licensed veterinary vaccines, and this is also a very generic technology, and the WHO has issued already DNA vaccine guidelines in order to help poor country manufacturers be able to manufacture DNA vaccines, even if they don't have something like our FDA to do the licensure. This also has been administered that will be entered into clinical trials probably next month using a device that's kind of called a gene gun that helps to propel the DNA, but many trials are just done simply by injecting with a syringe. Now, the last technology that I want to mention is recombinant viral vectors. And the reason I'm mentioning this is, in fact, China just got approval to start a vaccine, that clinical human trial using this. And this is using the outside of a different virus, in this case, an adenovirus that just causes the common cold, but putting inside a gene coding for the coronavirus spike protein. And so this trial also has gotten underway. So the summary is that we have 2 studies that have been started, for mRNA and also then for this vector. And then likely for DNA vaccines will start next month. But there are a total of 34 vaccines that are under development worldwide. Now, I've listed all of the ones here that have been adapted from the Fierce Biotech thing, but I'm not going to go through them. This is just for your usage if you want to come back and look at them. So just to summarize, I wanted to close by listing a couple of potential issues about the vaccine development. The first is, as I mentioned, we don't know whether targeting just the spike protein will be protective or not. The second issue is actually a bit more challenging, and that's that there's conflicting data about whether antibodies will last long enough to be protective to be a disease that continues to persist. And in fact, for other coronaviruses, which cause the common cold, it's been shown that in fact the antibodies seem to be of short duration and that people can re-get a cold even if they have high antibody titers against the coronaviruses. I asked the question of whether T cells will be needed or would they be deleterious. And then a vaccine often induces shorter-lived immunity, depending on the type of vaccine, than an actual infection does. So will the immunity from a vaccine last long enough? And then just finally, to raise the final questions are, this time we're actually kind of doing things a little bit different than most vaccine development efforts, where usually animal studies are done before you go into humans. They're now being done in parallel. And so the issue is whether or not we will find something in the animal studies that may indicate that we shouldn't go forward with the human studies, even if they look like they're safe. Maybe they won't be as effective in the animals or so on. There are unknown safety issues, and so this is something why it's important to continue the animal studies, but also to look for all safety parameters in the clinical trials. And then the big question, and Divya showed this amazing website, Next Strain, that shows how the virus is evolving. And one of the questions is, will a vaccine develop now against the current sequence of coronavirus 2 be able to protect against the virus that's present when the vaccine is licensed? And as you know, every year the flu virus strains have to be reevaluated. So the question is whether or not by the time a vaccine is made, it will be able to protect. So I will be happy to answer any questions, and I certainly hold out hope for a coronavirus vaccine, but we have a long ways ahead, and I'm just glad there's so many groups working on the many different approaches. Thank you.
Great. Thanks very much, Dr. Liu. One question for you. Everyone's in a hurry to get a vaccine done, and you can unshare your slides. Um, uh, Everyone's in a hurry to get this through, and it just does take time. Can you maybe share, your perspectives on how the usual vaccine testing process takes, how we could accelerate it, and what are the barriers to doing that?
So one of the things that actually has happened in recent years as a response to epidemics has been that that it is now possible to be able to do what's being done right now, which is to start the clinical testing before all the animal testing has been done. But of course, understanding this is a phase 1 safety test. And so that's actually I think the biggest change that has happened with the understanding of two things. One is that we just need to move things along as quickly as possible. But the second thing is now we have, in general, we actually have a fair amount of expertise with the different vaccine technologies. And so, for example, with DNA vaccines, it's really the same plasmid, essentially just you put in a different gene for whatever vaccine you want to make. And so there's a high comfort level with that particular technology that makes people and regulatory bodies able to say it's all right to start the phase 1 study. So that's been a huge change from 20 years ago before we had the earlier pandemics.
Great. And then the last question, you mentioned, we talked earlier before this session, that some vaccines have even gone through the whole process, but then been shown, I think in the case of dengue fever, to cause some very significant untoward side effects. So, the fight could be worse than the cure in some cases.
Yeah, that's actually one of the reasons I think that people are being so very careful about it. It's always been known that there have been specific challenges for diseases like dengue, and and so that's why I think that you will, if you just read the lay press, there's a balance of people saying, yes, we want to move ahead as quickly as possible, but we also will make sure that the animal trials are done so that we uncover if there are any issues related to COVID that were known about, for example, but not completely understood still for other diseases. So that is one of the reasons why the animal studies have to continue, as well as the pace of the human clinical trials still has to exactly never outstrip where we are in terms of our knowledge from the safety side.
Great. Thanks so much. We're going to keep you standing by. We're going to switch gears from vaccines to developing new drugs.